Welcome to episode 2 of COVID Weekly. My name is Craig Martin. I'm a professor and infectious disease pharmacist at the University of Kentucky College of Pharmacy. My co-host is Dr. Dave Fiola, who is an immunology expert, and we actually have a second immunology expert on today. If you're ranking our immunology knowledge, I would rank number 3 out of this group today. I'm just going to go ahead and lay that out there. I don't know who's number 1 and who's number 2. I'll let them fight that out. But Dave, who do we have today?
Thanks so much, Craig. It's good to be back with you on the podcast. We have Dr. Vince Venditto with us today, and we are excited to have him. Vince is an assistant professor at the University of Kentucky College of Pharmacy with us, Craig, as you know. Vince has a really interesting background, both in immunology, and also he's an expert in liposomal drug delivery. And it's his immunology background that we're going to draw on today and his expertise in that area. Vince came to us at the college after getting a PhD in chemistry at Texas A&M. And then he worked as a postdoc on vaccine development during his time at UCSF, the University of California, San Francisco. So Vince, welcome. We're really happy to have you. If you could open up just by telling us a little bit about what you've been up to with regard to the pandemic since it started.
Yeah, thanks, Dave and Craig. It's great to be here. I appreciate the invitation. So yes, as soon as the pandemic started, we everything was shut down, and we shifted gears pretty quickly. My lab is really interested in understanding the immune responses in different diseases, and we use different drug delivery strategies and to study the immune response. And one of those tools that we have in the lab is a high throughput robot, basically to evaluate blood samples. And so we pretty quickly set up a antibody testing platform in our lab so that we can screen serum samples from patients to look at the prevalence of COVID-19 or SARS-CoV-2 in the population. And so now we have a trial where we're using pharmacies as collection sites where we recruit subjects, we draw blood and take nasal swabs to look at the prevalence of SARS-CoV-2 infection in the population. And then we're also looking at how the virus is evolving within the population by doing whole genome sequencing. So it's really kind of an exciting time to be doing science where the whole world is focused on the same scientific project and fully invested in our results.
Absolutely, and because of that, and because of your other work, I know you've been extremely busy. So for the audience out there, Vince and I collaborate on a number of projects having to do with immunomodulation. And one of the drugs that we study is azithromycin for its immunomodulatory properties. And of course, early on, azithromycin was one of the drugs that was tried in COVID-19 patients, and we actually published a review paper on that. So I'm eager to talk some immunology today here with Vince. So Vince, could diving in here, could you briefly summarize for us the immune response to the SARS-CoV-2 virus?
Yeah, so, there's so much that we're still learning. And I guess when we think about the immune response to any type of infection, SARS-CoV-2 included, we typically think about the different players that are involved in this process. And we have T cells, and then we also have B cells. The B cells are going to generate antibody responses. And this is really what my lab is interested in looking at, but also generally what we're looking at when it's the easiest thing to measure. So that's typically what we're looking at with the large vaccine trials. And those antibodies are really generated to help prevent the virus from getting into cells and preventing it from spreading. And then the T cell response is doing something very similar in that those cells are helping to not only help the B cells produce antibodies, but also target any infected cells. And this is the difference between CD4 and CD8 T cells for that level of immunology. So the SARS-CoV-2 does induce a pretty strong response in most subjects. The response is variable, and normally what we see is three different phases of immune response occurring in these subjects. Uh, Typically, we have the early phase where you're getting the early immune response to occur, and this is the innate immune response. You're getting some early cytokine expression, things to stimulate the overall immune response that's going to eventually lead to those protective T cell and B cell responses. However, as the infection progresses, the the virus has certain proteins that can help change how our immune response is activated, and it can suppress these early protective responses so that it continue to propagate. And when if the virus has a chance to evade that early innate immune response, it actually gets to a point where the immune response becomes overactive, and it leads to things like cytokine storm and these more multi-system organ inflammatory responses where subjects progress to COVID where they need ICU treatment and supplemental oxygen.
Yeah, so as we've talked many times, it's that double-edged sword of the immune response where, we need it for proper clearance of a pathogen, but then in the case of SARS-CoV-2, the immune response then can drive some of that late pathology that causes severe disease. And so turning this then toward the vaccine, you mentioned this antibody response, and I think most of our audience probably knows that the antibody response is targeted to the spike protein, which is a protein in the virus, a protein that is essential to the virus in order for it to enter our cells. And we know that this is an mRNA vaccine. Can you tell us, how does an mRNA vaccine in this case, how does that work?
Yeah, so it's a great question. The With my background, as you mentioned previously, with liposomal vaccine design, this is really along the lines of my other passion in drug delivery and lipid based drug delivery. And so the way that these mRNA vaccines work is the mRNA is basically a genetic code, and I guess younger people who use Snapchat, that's what we think about when we think about mRNA, is that it's something that's presented to the cell and then it's gone pretty quickly after that. And so the mRNA vaccines, the genetic code is a sequence for the spike protein. It's formulated with some lipids, and those lipids help the mRNA get into the cell and into the cytosol so that the mRNA can do its job, and the spike protein can be expressed in those cells. And I mentioned that it's like Snapchat because once that mRNA is used, it's gone, and you don't the it's not visible to the cells anymore, or to the body in any way. It's destroyed. But that cell now has the ability to generate those spike proteins, and that's really how the immune system is activated when the mRNA vaccine is injected. So it's injected in your arm, so your muscle cells are taking up that lipid formulation with the mRNA, and it's those muscle cells that are also going to express those proteins. And once those proteins are expressed, then the B cells can come in. They can activate their antibody response, which is the one thing that we typically measure when we're looking at these vaccine responses. But they're also activating CD4 and CD8 T cells. The CD8 T cells are going to target those muscle cells specifically because they're going to look like they're infected. And the CD4 T cells are going to help with the overall inflammatory milieu to help the B cells and other cells provide an appropriate immune response to help drive protection. And it's the primary vaccine that really gives you the initial exposure, and the booster shot, which I just had my booster shot on Friday and was feeling pretty crummy afterwards. But that was really important because it told me that the vaccine was working and that it was doing its job. And it's that booster shot that gives you um, all of that extra immune response that helps to fight the infection if you're exposed to the virus.
So for episode 3, I hope, Vince, you'll come back and explain to us what Snapchat is for those of us who are over a certain age, and how maybe we can incorporate that into one of our studies. So I'm interested in what the vaccines have actually been shown to do, right? So we have a number that people see on the news that says, let's say Pfizer's vaccine is 95% effective. Can you help us understand what 95% means, and whether that means you're not going to get the virus at all, whether that means you're not going to transmit the virus to someone else, whether that means you're unlikely to end up in the hospital? What's our actual endpoint here?
Yeah, it's a great question. I think this is the importance of continuing to wear the mask, because we don't really know the what level of protection the vaccines are providing. What we do know from that 95% number is that so they took 40,000 people in these clinical trials. Half of 20,000, half of the clinical trial received the vaccine, the other half received placebo. And then they looked at how many patients who received the vaccine progressed to mild, moderate, or severe disease. And what they found was that um, vaccinated subjects were 95% protected. So compared to the placebo, all of the 95% of the cases that progressed to severe disease happened in the placebo group. So that suggests that the vaccine was preventing hospitalization and progression of disease. Now, what that doesn't tell us is whether or not somebody who is vaccinated um, is protected from infection, and most likely they're not, because we know that some people are still getting infected. We just don't know if it's a decreased rate of infection. And the other thing we don't know is, if you're vaccinated, can you transmit to other people? And so, data that I just saw yesterday that was published in a preprint, they looked at a cohort of subjects in Israel. Israel has one of the most advanced vaccine rollout programs, where they've now immunized 77% of subjects who are 60 years and older. And what they found was that it looks like, since they rolled out the vaccine, that subjects who are 60 and older have lower viral load as determined by PCR. So it suggests that data there. That clinical trial is not properly controlled, and there are some questions that we still have to answer. But it does hint that there is not a complete elimination of virus, but there is a decrease in the amount of virus that's in these patients. Obviously, there's a lot more work that needs to be done, but I think it really does set us up very well for the value of these vaccinations. But until we have all that data, I think it's important to continue wearing a mask so that we are not spreading it to the community around us.
Yeah, absolutely. That's well said. And for what it's worth, I really do like that Snapchat analogy. I don't really know how to use Snapchat, but I do know that whatever message is sent is gone very quickly. And I know there's some fear out in the communities about the vaccine, because when you hear mRNA, that sounds a bit scary in terms of what that could potentially do. But I think it's really important to emphasize the fact that what we do know about mRNA is that it does its thing in the cytosol of the cells, right? So it's translated into protein in the cytosol. So at no time will the mRNA enter the nucleus of any of our cells and cause any kinds of changes there. So that's good. And you mentioned about what 95% means, Vince. So of course, one of the concerns and big questions that everybody still has, and is really at the crux of things, with the vaccines is that durability of protection. And, how long are our antibodies going to last? What do we know? What's the latest on what we know about the durability of the antibody response?
What we know right now is only about 8 months of data since the clinical trials were initiated. We do know that the people who are infected have a pretty variable, the people who are infected without vaccination have a pretty variable immune response to the virus. And the variability there does not necessarily lead um, to long-term protection, but the vaccines do appear to elicit a pretty strong response. It looks like it's pretty focused and it's persistent for some amount of time, at least for 8 months now, is what they've measured. I think there's a few interesting pieces of data that have come out recently. One is that the mRNA vaccines are a little bit different from other vaccines in that they do induce germinal centers to form. And germinal centers are really important for generating that immune memory that's going to keep you protected if you are exposed to the virus later. Um, and because of that memory response that is induced, there are projections that the vaccine will induce protection for at least a year. Now, obviously, there's a lot more work to be done to actually track this, but I think it's a really good sign that germinal centers are being formed with this vaccine, and we are able to see the persistent antibodies and memory responses.
Yeah, I'm glad you bring up memory because I was going to go there next. I think it's important to understand that even though our antibody titers may wane in our bloodstream, that memory response should still be there, hopefully. Some of the work that we were talking about earlier today, Vince, as you know, on SARS-CoV-1, actually shows that even without memory B cell responses, decades after a patient is infected with SARS-CoV-1, those patients still had memory T cell responses. And so those T cells, as you were talking about earlier, have the ability to then stimulate B cells and in these germinal centers to then start producing protective antibody. It might be a little bit delayed. Versus having the antibody already present, but hopefully there'll be some protection there. But if that's the case, what do we know about how that memory can impact transmission? Do we have any idea of really its impact there?
Yeah, it's a good question. I think the one the if we look at natural infection with SARS-CoV-2, Shane Crotty just put out a really interesting paper looking at immune memory induced to viral infection and looking at memory responses over 8 months, and found that early on, the T cell response is really what's driving the protection from progression of disease. And that T cell response doesn't necessarily match with the antibody profiles that we're seeing in those subjects. And later on, the antibody responses are uh, providing a lot more protection. But long term, it's the early T cell responses that are leading to that appropriate memory response that we see in those subjects that are infected. And so I think that really the memory responses are key, but the variability in the memory response to the virus, although it's happening in a number of patients, it is variable, as I mentioned. I think the advantage of the vaccine is that as we discussed, it's really it's only a single protein, so we're typically, well, at least with the Pfizer and Moderna vaccines, it's only the spike protein. So when we're inducing those immune responses and we're generating that memory, it's really we're generating memory to a single protein. So the immune response is much more targeted and it's not distracted by all the other proteins that are present in the virus that can, I think, distract the immune response away from being a protective response. And we know that viruses use various strategies to evade the immune system and try and limit the amount of destruction the immune system has on the virus. So the fact that we have a vaccine that's targeting a single protein, I think, is a huge advantage in making sure that we can get out of this pandemic on the other side.
So, Vince, there are a couple of sort of related topics that I want to sort of unpack a little bit. You mentioned earlier the sort of delicate balance of the immune system and its necessity to fight off pathogens and its potential swing too far in the other direction to cause some of these severe consequences that we see, whether it's in with SARS-CoV-2 or with bacterial infections or fungal infections. It tends to happen with multiple different kinds of infections. And so I was fascinated in your paper with Dave that was mentioned at the top of the show. You sort of warn clinicians that they really need to be thinking carefully about immune modulatory therapy. And this was in reference to azithromycin. Can you talk a little bit about that balance a little bit and why a drug like azithromycin, which was used pretty significantly at the beginning of the pandemic, why that might be a little bit dangerous or a little bit, I guess, premature to begin using it in that manner?
Yeah, I think that's a great point. I mean, it is something that, it got a lot of press early, and that press was not very positive because of the context that it was used with hydroxychloroquine. But I think, when we think about the balance of the immune response, it's really that you want to have a productive immune response early on. As I mentioned, it's that T cell response early on that's going to lead to that memory and prevent progression. But so if you're giving a drug like azithromycin that has this immunomodulatory property, you can effectively blunt that T cell response and blunt the pro inflammatory response that you need to help fight the virus. And so, if you give azithromycin at the wrong time, like early on in infection, you can potentially prevent a person from mounting their own immune response and preventing them from fighting the virus on their own. But then, as a patient moves from that early stage of infection to a later stage where they have this really overactive response, that's really the place where targeting immune suppression and immune modulation is really key. And what they found and what clinicians are doing is administering different immunosuppressive therapies in that later stage once patients are progressing to the ICU, and that's really to blunt that overactive immune response and blunt the effects of those cytokines. So I think azithromycin fits in that same paradigm where treatment late in infection can actually help subjects improve. And azithromycin has some really interesting properties where glucocorticoids would suppress the whole immune response, but azithromycin actually helps modulate it instead of suppressing the whole immune system. It helps basically retune the immune system so that it's not an overactive pro-inflammatory response, and it's promoting repair rather than inflammation.
You led me straight into the next point, which is, many of these patients with severe COVID-19 are receiving dexamethasone in the ICU. I remember from my clinical days taking care of patients with other infections, this was always a significant debate amongst the medical team. Do we, in this patient with infectious disease X, begin corticosteroids? And on the one hand, you want to make sure that you are not blunting their initial immune response, or overshooting the blunting of the immune response, maybe is the best way to think about it. So thank you for that insight.
Yeah, absolutely. I'll chime in on that too. As we've seen the therapeutic guidelines for COVID-19 kind of progress over the months, now if you look at, the immunomodulatory drug dexamethasone really is only recommended in a very narrow patient population when they do get in that situation with progressive disease, because just like Vince and Craig both mentioned, we really don't want to counteract that immune response that's responsible for clearing the virus. We just want to control it once the immune response becomes a huge element of the pathology. And as we know, that drives some of the late pathology in the patients who are really reacting in a severe manner. And speaking of immune reactions, I'd like to ask a kind of a separate question, Vince. And I know we're going to have an upcoming episode that focuses in on some of the disparities in terms of social determinants, between certain populations. So, we know from early data that Black and Hispanic populations, for example, have worsened outcomes. So we're going to talk about that in an upcoming episode, I believe. But I wanted to ask you, there's an immunologic element to that difference as well. Could you take us through that?
Sure. So the term that we commonly think of is weathering, and really what that refers to is a constant stress that's generational, and it's certainly, there's evidence that it's carried down from generation to generation, and that it simply dealing with uh, different situations that put certain populations at increased risk for health disparities, and these are the health determinants um, of the health determinants that lead to these health disparities. So things like socioeconomic status and redlining and the institutionalized racism that really leads um, to what is collectively known as weathering. And that weathering is really there's epigenetic changes that are occurring. And then in the immune system, there's increase in stress hormones. And so anytime that anybody is stressed, you'll see certain immunologic things that happen. I know that when I was in graduate school, I was writing my dissertation, getting very little sleep, probably not eating or exercising appropriately. And I was starting to have some immunologic side effects of me basically not taking care of myself. And um, I could certainly start eating better and exercising and sleeping more. And that helped reverse some of the symptoms that I was seeing. But we know that a number of people in our community can't very easily do that. And those stress hormones are really elevated their whole lives. And so those stress hormones are, cortisone, cortisol, these type of hormones that are expressed to partially to activate specific immune responses. However, when they're expressed for such a high at high levels for such a long amount of time, you actually basically lead your immune system to go into exhaustion, and so your T cells stop functioning properly. Your all of your immune cells stop functioning properly, and it's that constant exposure to these stress hormones that actually end up going from a overactive immune response to basically a suppressive state where your immune system just can't respond to viruses and different um, insults the way that it really needs to. And so, we still have a lot of work that needs to be done to figure out why we're seeing the health disparities that we are seeing and what the exact cause of these health disparities are. However, it's quite clear to me that if you have elevated stress hormones for your whole life and you are um, immune suppressed, that if you're infected with SARS-CoV-2, you're not going to have the immune response that's necessary for you to fight the virus, mount the appropriate T cell memory, and unfortunately, it's these populations that are having worse outcomes.
Thank you for that, Vince. And I think that's something that we don't often think about is the long-term consequences of what we usually consider non-health stressors, right? But how much of an impact that can have on generations of people. And I appreciate that insight that you have. So I'm going to follow up with one final question, and I kind of want to ask you to put on your wizard's hat and get your crystal ball out And I want you to put yourself in late August, early September 2021, later this year. And I want you to tell me if you think we're going to be going to football games or if you think we're going to be taking our masks off inside. What do you think society is going to look like? And I'm also going to ask you when you're going to go eat in a restaurant again.
That's a good question. I think I have been asked this question a couple times, and I think it's important. Certainly we have a very Lexington and Kentucky centric view of how we're thinking about these things, and I think that perspective is important. But I'm gonna take a slightly broader view to start, and then I'll come into Kentucky. And that is, if we look, at the basically we need to get to herd immunity, or what I prefer to call community immunity, and that's really the point where enough people are vaccinated to have protection. And so if we look at that at the Kentucky level, we're getting there. We're certainly vaccinating a number of people a day. But if you look around the world, it's projected that Africa is not going to have vaccinations until 2024. And so you think about um, us getting back to normal life, and to me, normal life would be traveling and certainly doing what I'm doing in Lexington, but also going to conferences around the world and going on vacations. And if a significant part of the world doesn't have vaccines until 2024, I don't see us getting back to so-called normal until certainly after the world is vaccinated. And so I think that there's we are unfortunately, I think, going to be dealing with this for a long time. Now, so travel restrictions are going to be limited. That means, shipping restrictions are probably still going to be impacted. I think, we're struggling to get pipette tips in the lab right now because of product shortages and things like that. I think, um, so I think we're going to be living with this for a while, and it's important for everybody to get vaccinated when their name gets called, so that we can begin to get back to some level of normalcy. I think so. To your question about when I'm going to go to a restaurant, I think I would want to see that Lexington, Kentucky, certainly has the majority of the population vaccinated, and I would like to keep travel restrictions and all these types of things in place for the foreseeable future, because I wouldn't want to have people from some of these other countries, or even surrounding states, if they don't have the appropriate um, conditions in place to prevent spread. Um, and what we're dealing with right now is all these new variants that are popping up, and we know that they're popping up because of a lack of mitigation strategies. And so until the world is on the same page and we have all those mitigation strategies, we're not going to be dealing with, we're going to continue dealing with new variants popping up, and those variants can certainly get into our communities, and we may or may not be protected with these vaccines. So certainly a lot more research to go forward to figure that out. But I am not going to probably be going international travel for more than a year, and probably not to a restaurant for I don't know, maybe another year. I don't know. That's a tough question.
I think I collectively just heard our audience screaming at their headphones, saying, "Please don't let this be true." But I appreciate the realism, because I think you're right. It's not an easy fix. It's not a short-term fix.
Well, and to bring us back, the fix lies within our ability to understand the immune response and to take advantage of it with these vaccines. And so, like Vince just mentioned, everybody getting vaccinated when their opportunity comes up. Everybody continuing to practice social distancing and wearing masks to keep those numbers down. And really, when the vaccine, as Dr. Dutch on our last episode mentioned, when that vaccine starts impacting transmission and also impacting the severity of disease, hopefully we'll inch back toward normalcy. Well, Vince, I want to thank you once again for coming on. It's been fun to take a bit of a deep dive into the immunology of this virus and of the vaccine specifically. So we really appreciate your time.
Thanks for having me. It was great.
Thanks, Vince.
Thank you.